title for which is Role of Artificial Intelligence, Cardio-Oncology and Challenge in Cancer Treatment. So I welcome our session chairs for the day. We have Professor Dr. Subrajit Biswas, Director, Amity Institute of Molecular Medicine and Stem Cell Research and Convener, IGCC 25. Our second session chair for the day is Dr. Puniti Mathur, who is working as a professor at Amity Institute of Biotechnology. We welcome you, sir and ma'am. I would now request our session chairs to please introduce our speakers for the day.
Thank you very much. So very good morning to all. Nice to see some students in the hall now, and we'll start the first session. Our first speaker today is Dr. M.K. Datta, the Director of Amity Center for Artificial Intelligence and Dean of Students Research at Amity University. He has expertise in AI and advanced research methodologies. He's also served as the Director of the Center for Advanced Studies, Lucknow, and is a visiting professor at PRNO University of Technology, Czech Republic. Dr. M.K. Datta is maintaining a very state-of-the-art AI center at Amity University, and I'm sure you're all going to gain a lot through this talk into the inroads of AI into medical research. So Dr. Datta, may I request you to please start your session?
Okay, good morning. My name is M.K. Datta. Thank you, the organizers, for inviting me for this talk, and thank you all the session chairs and my colleagues for inviting me, and great standing in front of you, and all my students, colleagues, friends, delegates. First of all, best wishes for this great conference that you people are attending. My role is different. I'm an outlier. I'm not a cancer scientist. I'm not a biologist. I'm a computer scientist, or specifically you can say I'm an AI scientist. But then in the current times, what you have seen around, AI has already become an integral part of all sciences, all studies, all programs, all routine activities of our life. So cancer biology or any biotechnology cannot remain isolated. And of course, all students and my colleagues, you all appreciate that artificial intelligence is a futuristic technology which is going to take you forward. You cannot live in the futuristic, in the what I can say, in the years to come, if you are not conversant with AI technologies, using them, most likely you'll live one step back. My talk is about artificial intelligence in cancer diagnostics. So what I'll try to present, I'll just show you some use cases, which is published, which is in in public domain. You can always go back and read more about it. There is some shortage of time, so I'll try to keep things simple and try to push and my slides. Whenever I start a talk on AI, because AI is a buzzword and a lot of noise going around AI, I try to show this. A timeline. In fact, AI is nothing new. AI is way back 1954. This year, Geoffrey Hinton, who got Nobel Prize for creating deep neural networks and artificial neural networks, the theory was in 1974, for which he got Nobel Prize in 2024. So the timeline, if you can see, there was AI in 1954, in 1960s, 1970s, 1980s, and if you look into this timeline, when once comes 2018, there was 2018, there was a model given by Google DeepMind. Google DeepMind is one of the top companies of United States, where there was an Indian scientist. His name was Ajit Baswani, okay, and there was a Punjabi girl named Nikki Parmar. They had given a paper called Attention is All You Need, 2018, I think 2017, December. And that model, that architecture actually revolutionized the world. It changed everything. Like what I can say, let's say, I can say 2018, AI becomes unlimited powerful. The power of AI becomes unlimited, infinite, and AI is capable of doing, almost everything. Of course, there are bottlenecks like compute power and structured data availability. But then the architecture, the context memory, or the understanding of AI has really taken a big step in 2018. And I think all of you have seen in the last 7 days some amazing news going on. Have you seen, students? Do you know what is going on? The US AI market has crashed. $2 trillion, $2 trillion the companies have already lost in US, out of which I think 1.5 trillion is only one single company, Nvidia. So a lot of amazing things going on. There's a Chinese company. Yesterday I was actually in national television talking about all these things. There's a Chinese company called DeepSeek who has intervened, and they say, their model is completely open source. It's completely free, and they can be trained with less number of parameters, giving young students like you a lot of leverage to train models and use AI for your own, applications. So this is an important timeline curve always I show, and indicating that AI has become super powerful from 2018. This is a quick glimpse of our center. Our center is in E3 block, ground floor, room number 16. All of you are welcome to come here, come in our center, discuss your ideas. We are always looking forward to mentor students, collaborate with faculty in whatever ideas you have. And in the current times, the modern times of AI, when generative AI and transform models are so powerful that any idea that you have, we can help you. And you can see we are a state-of-art lab. We have 10 petaflop of computing power, 640 GBs of RAM, and we have DGX 800 cards, which is good enough to compute any amount of data. A quick glimpse of our center. We do a lot of research like machine learning, deep learning, Gen AI, the core research for computer science, and also for anyone who is an applied scientist. Most of you are applied scientists over here. You are a biotech student, or you are a cancer biologist, or you are a cardiologist, or you're a liver specialist, or whichever field of biotechnology, civil engineering, mechanical engineering, aerospace, space science, whatever robotics, whatever area you are, we're doing a lot of applications, and students and faculty are always welcome to collaborate, join us in this mission of artificial intelligence. Okay, this is where we are. I'll quickly try to, this is a summary of what I'm trying to discuss. Although I think I have more than 250 research papers in artificial intelligence in healthcare, I think around 250 or 260. So here, because it's a cancer-related event, so I'll try to keep my talk in breast cancer. So some of the cancers in which we have worked, I'll just try to explain you in a very simple manner so that you understand. And my talk becomes effective only if you understand and get motivated in doing stuff like that. And all the work, whatever I'm going to show you, I'll show you 6 use cases. And all these 6 use cases are actually published in research papers. They're available online, or you can always come back to me if you cannot find the full paper. First one, I talk about a very interesting application of breast cancer. Now, breast cancer, I think WHO, I'm not a cancer specialist, once again I repeat, I'm not a cancer specialist, I'm just an AI scientist. I collaborate with cancer specialists and doctors in developing AI apps. This work was in collaboration with Dharamshila Hospital of Delhi. So we had some data. I tell you it's like that. WHO has already given you some kind of advisories that beyond a certain age, a woman should do breast cancer tests, I think twice, once in 2 years or something like that. Now, the breast cancer examination is like a mammogram or an imaging-based method, which may be costly, or and there may be situations where you do not have access to these kind of equipments in tier 2, tier 3 cities. So the entire research was can we actually predict breast cancer with some blood biomarkers? So that was the study. So the data was collected from Dharamshila Hospital. So this is the pipeline. You can have a look. This is the pipeline where you can see the entire pipeline. We had collected data. So there were actually 3 different these are the biomarkers. You can see, I do not know much about all these biomarkers. For me, this is just a data. For me, this is just a number. So first of its kind, AI model integrates 3 parameters, beta HCG, PD-L1, and AFP biomarkers. I think you can understand, you can do a simple blood test. It will cost you maybe 100 rupees or 200 rupees. You get these values, and based on these values and also some demographic values, some age and some other symptom that you're going to input in the model is going to return you a diagnostic probability of how close are you to breast cancer. So this is the pipeline. Now, if you really see, the data was collected. I think we had 1200 and data for each classes. There are, I think you can see there are grade 1, grade 2, grade 3, grade 4 and control subjects. So we had a database of around, I think, 6,000 data which was shared by Dharamshila Hospital, and we tried, and because data cleaning and getting the data structured and to find the the spatial dependencies and the temporal dependencies in the data is important. And also as you study data science, you will see finding the right machine learning model that actually fits in to look into the spatial dependencies or the temporal dependencies of the data and train it accordingly for giving or solving a task is sometimes complicated. So this project we did for, it took a lot of time because that machine understands a data lingo and not necessarily that the temporal dependencies which a human mind can interpret, machine cannot, because machine simply goes by patterns. This was not a reinforcement learning, by the way. It was clearly a deep learning model we tried. Anyway, so I proceed with that and I show you this is the pipeline. You can see there was data collection. The data was collected, then it was labeled by the cancer biologists of the Dharamshila Hospital. There was medical experts who labeled it. Then after labeling, we handled the data imbalancing part where there were many things like missing value interpretation. We tried to remove the imbalance, and also then we tried heterogeneous classifiers for biomarker data analysis. And what we saw, let's say a linear kernel. Normally, machine learning we use a linear kernel or RBF kernel, or there are some standard kernels which works. Unfortunately, it was not working. So we tried an ensemble method. Ensemble method is a voting preference probabilistic machine learning model. Here I show you. And there we saw that the inclusion of these biomarkers, these blood biomarkers, included with some demographic details like age of the patient, height of the patient. There were certain more things which apparently seems to be more important. Discriminatory in deciding whether they're close to breast cancer or not. So this is the pipeline. You can show the classification algorithm we tried. We tried multiple actually. We tried actually whatever possible, and then we found that the ensemble method of machine learning gave us a very good results. The app is deployed. You can go to an Amity Center for Artificial Intelligence. We have a website. In this website, you can try the app. It's free. Anybody can use this, and we have made it open source. So here I'll just try to summarize the thing. The step 1, we collect data, label the data. Step 2, we try to clean the data, make it as structured as possible. The interpretation part, labeling it, handling the missing values, handling the imbalances, and then we try for training the data with different models. Once again, students, remember that any machine learning model, actually, when I say artificial intelligence, it's a misnomer, because anything which is artificial cannot be intelligent. You agree to it, right? Anything which is artificial cannot be intelligent. So a lot of mathematical operations happens like differential calculus, linear algebra, and a lot of things. Actually, whenever, let's say you work with ChatGPT, you give a prompt and you get a lot of response, and people start thinking this fellow is intelligent. Do you really think it's intelligent? Definitely not. Let's say ChatGPT 4, which you're currently using, is an 80 billion parameter model, which effectively means that whenever you put a data inside that, there are 80 billion parameter points which are affected, 80 billion differential calculus, 80 billion linear algebra, 80 billion matrix multiplication, 80 billion convolution operation happens and returns the value. So it's purely mathematical based, so statistical operators are used in that. So if you see here, after doing this, we got an accuracy which we have 97.93. It was one of the best accuracies that we have got after doing 5-fold of cross-validation. I've given a screenshot of the paper published. The paper was published in one of the Elsevier journals, and if you want to read more about it. So I conclude this use case saying, for any lady in a country like India, where they may not be having access to medical imaging to test the breast cancer diagnostics, we are suggesting an alternate way, a cheaper way, where you do a 100 rupees or a 200 rupees a blood test, you get the values, enter your demographics, and punch in, and it will return you how close are you to grade 1, grade 2, grade 3, grade 4, and that's how the doctors have tested it, in Dharamshila Hospital, and we have got an accuracy around 97.93% in this. Next, this is cytokine genetic variants and socio-demographic characteristics as a predictors of cervical cancer, machine learning approach. This is a little different. The previous case was, I'll go ahead with this also. So this is also a published paper. So here I will have to acknowledge the King George Medical University in Lucknow. So the data was collected from King George Medical University in Lucknow from cervical cancer patients. So once again, after discussion, I have to acknowledge Monisha Banerjee, who is a clinical biologist in King George Medical University, and few more doctors. They are included the author of the paper. What there was, initially the doctors, they suggested few parameters, in your language, biomarkers, which may be responsible or discriminatory in deciding the probability of occurrence of cervical cancer in women. So here you see the risk factors included are categorically of two categories. One, the histopathological parameters. One is the histopathological parameters, that is the biomarkers, and second was the demographics. So the idea was, can we combine the discriminatory values of demographics of a patient with the histopathological parameters, the cytokine genetic variants, which once again you have to do a blood test, maybe 100 rupees or 200 rupees, a blood test where the values comes up, and can it be discriminatory to decide the prediction of cervical cancer in subjects. So here you see once again risk factors, socio-demographic factors like age, place of residence, whether it is urban or whether it is rural, educational status, educated. We put some, if you want to know more about it, you have to read the paper. It's available online. Socioeconomic status, parity, age at the first full-term pregnancy, menstrual cycle, whether it is regular or whether it is irregular, menstrual hygiene. We give some options like what kind of menstrual hygiene you are maintaining, what kind of contraception you use, smoking habits, and then the histopathological parameters, the biological value, like which I have mentioned over here. The combination of this, when we collected about 7,000 patients from King George Medical, they had a long history. Of course, one argument against this, maybe it's limited to North Indian population only, because King George Medical University concentrates mainly in UP, the North Indian population. So the data was collected from North Indian population only. And using these demographics and the histopathological parameters, we once again tried a lot of models. So this paper is little older, 3 years old. We worked on this around, but of course we are still working to improve some of the things in this model. But what I'm trying to show you here is that once again we tried all the possible machine learning models using these biomarkers to see whether this fellow can predict cervical cancer or not. We got, I think, 92 or 93%, I think, is it mentioned somewhere? Most likely 90% above accuracy we got in prediction of cervical cancer using this habit. So it's not only a diagnostics, but if these habits, if you have this kind of pattern with the social demographics and the genetic variants, you are close to cervical cancer. So this was also tested by Monisha Banerjee, who was in King George Medical University, and the results were reported. The paper is online. I've given you a screenshot of the published paper. I think it's published in Computers and Biology. This one's in top journal, so you can read more about this if you are interested. So interestingly, once again, you can see, to sum it up, this use case is like cervical cancer. Otherwise, it's a pap smear based. I don't know, the conventional method may be costly, and I'm not sure, you people know it better. Here, it's an easy method of doing a blood test, putting the marker values and some demographics, and checking for yourself how close you are to cervical cancer. Maybe now you have understood the pipeline. I think there's some shortage of time, so I'll push the next slide a little faster. The same story with glioma. So glioma is a neural disorder. I'll run through this slide. So glioma is a neural disorder. I think it's a brain cancer or a neural cancer, in your lingo. So here the symptoms I have mentioned, and the paper which was published, I have put it as a screenshot. This was also in Computers and Biology and Medicine. So ensemble-based glioma learning approach for prediction of glioma and multi-grade classification. So here I need to acknowledge AIIMS Bhopal. And I think there was one more hospital. There were two hospitals in Bhopal, from which Puneet Gandhi was the doctor who was collaborating with us in this problem. So these are the clinical things, like what causes and what the risk factors of glioma. And the medical imaging, this is how the MRIs looks like. So we are not going in the conventional method, but once again the same story. The repeat of, it's similar pipeline that we've used in the previous two, like the cervical cancer, the blood cancer. We try to find some histopathological parameters. Okay, we try to find some histopathological parameters. I've mentioned the histopathological parameters here, like HTRT. Then you have got YKL40. I've written this histopathological parameters. I think with the blood test you can get that, and then after the gold standard was done, doing the conventional test, the database was prepared. So we had the database of these grades of glioma. It was a multi-grade classification, but the doctor insisted that we should go for the grading, not only a yes/no problem, but also the grading of glioma. So once again, you can see the entire pipeline. We did a five-fold cross validation, and we tried to train it with all possible machine learning models, and we found that the ensemble-based method, which is based on a voting preference, ensemble method actually works when there is more of what you can say non-linearity or complexity in the data. If you cannot find an easier model to represent the data points given in a plane, any dimensional, let it be two-dimensional or multi-dimensional planes, you may not be in a position to actually find a model that segregates points. So ensemble methods becomes very handy if the distribution is too complex to handle, where the linear models or the like radial basis function models do not work. So that's the pipeline. So it was I think as I mentioned, it was a voting preference done on statistical operations, so combining the individual model predictions, and we did the ensemble method for finding out, and we got good results. I mean, you can see this is the entire pipeline, the histopathological analysis, the reports. From the reports, we generated the control and the glioma case. I mean, from the gold standard data, the gold standard the data was generated. The pathological characteristics detected by glioma, which was transferred to the machine learning model as the pipeline of the biomarkers, which was trained, and accordingly, the grades like grade 1, grade 2, grade 3, grade 4 of glioma was done. So this is how the parameters look like in our study. The mean, max, and the average values are mentioned for 1, 2, 3. The grade 2 control subjects, grade 2 glioma, grade 2, grade 3, and grade 4. And the performances of the machine learning models you can see here, we tried a lot. And the last one was the proposed one, which actually is returning a very, of course, if you, in a machine learning lingo, if you understand, if you look into this, the other models did not perform well because of the too much of non-linearity and overlapping in the data points, where you can see the ensemble method is doing better in comparison to the others. So these are the results. The paper is already in public domain. You can read more about it. Okay, the next use case, maybe I'll discuss 2 more use cases. This is the next use case. Here we talk about polycystic ovary syndrome using ultrasound. The last 3 use cases that I've shown you was purely on biomarkers. We did not use any computer vision model in our input to generate any kind of decision. Here we come. So this is attention-based multi-scale deep neural network for diagnosis of polycystic ovary syndrome using ultrasound images. Once again, the paper is available online. You can always read about it. So this is about ultrasound images. As I mentioned, in 2018, when the model came, the transformer model came, there was an amazing model called transformer model came, which actually gives you an interesting concept of attention. Now, what is attention? Let me a little bit explain. Let us say from a radiologist point of view, let us say a radiologist is looking into this ovarian ultrasound image, there will be positions, there will be portions of the image in which the radiologist is concentrating to take a decision. It's not that for example, let's say I look at a face and I say this is a person. There are something like the eyes and the lip contour and the jawline. There are what I can say, more discriminatory points in any image which needs more attention let's say I know a person, let's say I know you and I know your name. When I say you are Mr. John, it actually means that I recognize your lip contour, I recognize your eye pattern, I recognize your jawline, I recognize some of the features which is detrimental or discriminatory in identification of yours. That's called as attention, not necessarily. For example, if I only expose my face to you, this portion, you may not recognize me, right? But maybe if I recognize my eyes or my lip contour or my nose or some jawline, maybe you can recognize I'm Kadata, right? So this concept being translated, the attention mechanism, the points at which you need more attention in becoming detrimental or making a decision, was introduced in 2018. It's called the attention network. Okay, human vision works in attention network. There are points of discrimination where we put a lot of attention in making decisions. So we used I'm not going to the nitty-gritties of attention network. You can read more about it. But attention network is a super powerful network. Okay, it will not analyze the entire image, but it will focus on those images, on those portions of the image which have discriminatory patterns. We did that. I wrote about the images that we did. Of course, this is a pipeline. Of course, it was an open source image which we used. And this attention-based multi-scale convolution neural network, the architecture that we used, with dilated convolutions. Not sure if you know the dilated convolutions. The mathematical operations becomes much easier. We are focusing the field of view of the convolution where you're running a windowing operation across an image to pick the convolution and then activate it through the layers. you are dilating it, which effectively means the number of computations gets lower, that you are triggering less number of parameters in training the model, and we use this dilated convolution there. And the results actually shows that the dilation is decreasing the number of parameters used without impacting the overall accuracy of convergence of the model. The integration of this multi-scale characteristics achieved by attention mechanism, which enhances the importance of features with significant channels. We did a channel attention here also to identify in which channel, which are the points of more attention. So it's not only, I'm not able to explain. Maybe let me try a little bit better. When you look into an image, it is not only the the spatial representation of that particular point, but also the location, where is it located? Am I able to explain? The spatial as well as the position of that image. There's something called as positional encodings, etc. So when you make a decision looking into something, it is not only, let us say, your eyes. You know that in a face, the eyes should be like this, and the eyes should be located in a particular position of your face. So we not only concentrate on the spatial representation, but also where it is located. So this is how the attention mechanism works. So you essentially see the contribution in this computer vision model is that we used an attention model. I have mentioned over here that I've also shown a global leveraging pooling of this attention network where the attention scores are generated, and you can actually focus the attention map that comes out. You will see there's a heat map generated after the model is done, where you can see the points. For example, you're doing a tumor analysis, or you're doing a lesion analysis, or you're doing a hemorrhage analysis. The points of tumors I have time? Okay. And the tumors or the lesions, which are of more importance for us for making a discriminatory decision, okay, those points in the heat map will appear brighter. So that's how attention model works. And this is the architecture. You can see the feature extraction architecture between these layers. Sorry, you can see between the convolution layer and the fully connected layers. I have brought an attention mechanism, and the channel attention block generates channel attention matrix to analyze the input features map in interchannel interactions and minimize feature redundancy across the channel. We are not trying to put more weight to those points. Let me explain. Let's say my face. You want to do a face recognition. I do not want to put too much of weights in this portion of my face, which is otherwise not too much of edgy or too much of discrimination, where you want to put more weights on my eyelids or my lip contour, where you want to do that. So similarly, the attention network will give more weights to those points which are discriminatory in making a decision of a tumor. Or in this particular case, it is polycystic ovary syndrome in ultrasound images. And in fact, use of this, the novelty or the contribution over here is use of this attention model, so that we are focusing. Just like a radiologist looks at different locations, for making a decision, we are doing that using this attention model. So this is the confusion matrix and the ROC curve. We have very good results that is published. I have also given you a screenshot of the paper that is published. You can read more about it. And in case it was published by one of our BTech students in Belgium, so we are very proud of the student. I think Suzanne. Yeah, Suzanne. Suzanne is one of our BTech students who presented this paper in a conference in Belgium. So many congratulations to my students, Suzanne, if you're maybe two more. Now this is I call it as a FCCS net. This is a breast cancer, but in this situation we're not using a biomarker for training the model, but I'm using the histopathological images. So here you see, okay, I'll show you here. We use public databases. You people know, I think, know better than me. We have some public databases, so we use 3 public databases in training our models. The histopathological images from which we want to extract discriminatory features to do that. I'll tell you the contribution. Our contribution over here, look into this. What we have done, if you understand this convolution neural network, the point is, if you look into this images, which is distinguishing between yes or no, benign or malign, the radiologist is being trained. He knows to look into an image and give you a decision. So what we have done here is that we have used a base model. We have used actually ResNet as our base model, ResNet 18 as our base model. So there are 18 layers of convolution happening. But our innovation and our contribution in this part is that we used, you can see we have used multi-level attentions in between these layers, and also we have used some residual connection to ensure that the gradient vanishing problem do not happen. not sure if you understand. Gradient vanishing problem is a problem that occurs in a backward position when you're using a dense layer, let's say 5 layers or 10 layers or more number of layers in training a model. I mean, there will be more number of parameters and neurons involved in training process, right? So the contribution here is that and I have also used multiple attention networks. You can see there are multiple attention networks over here, which essentially is allowing at level by level, I mean at mid-level features and the low-level features, to concentrate only on those particular features which are important for making the decisions. Okay, and to also once we did that, what we saw that the introduction of the attention was leading to some kind of introduction of attention was leading to some kind of gradient vanishing problem, and the entire backpropagation pipeline was disturbed. So we had to use residual networks, which preserves the gradient for doing the backpropagation. So it's already given in my, if you want to read more about it, you can see this is the proposed FCCS attention model, and this attention model was actually designed by customized for this particular histopathological cancer detection problem. So, and it was tested, it was tested in a good number of databases. Of course, there are public databases and report. I think this paper was published in Scientific Reports in Nature. And here you can see, this is what I was trying to explain to you. When you do the heat map, when you do the heat map of the attention network, you see the bright portions of that images are those points of attention which actually is detrimental in making the decisions. Okay, this is the beauty of your attention network introduced in any convolution network pipeline. Oh, it was published in Biomedical Signal Processing Control. Okay, and results are very favorable. I mean, you look at the confusion matrix and the model convergence. We have got very good model convergence, and also we can see very good this one, the confusion matrix, the accuracy in the confusion matrix. And also interestingly, if you see the flops, the computational complexity, just compare with ResNet 50, DenseNet 201, DenseNet 121 and VG16, just see the number of parameters we have used. 2.2 million parameters we have trained. So that's one of the biggest contribution which the reviewers also appreciated, that we could do it with minimal number of parameters. So 2.2 million parameters, where others are much more, 5 times or 10 times more. So that's a contribution. See, as a student, as a researcher, it is not only that you give a robust model, but try to keep the compute as minimal as possible. Maybe I should quickly. So similar story is about pancreatic cancer. We used urinary biomarkers. Take a urine sample, generate the parameters from them, and train models to see how accurately the pancreatic cancer can be detected. I'll skip this. You can read more about it. This paper was published in Springer Nature Journal. Yeah, maybe this is my last 2 slides. I would like your attention here, because this is something which we are doing new, and we are super excited about this work. Whatever work I have shown you previously, essentially they are a little bit outdated if you look into the context of AI in the current times. So, but this what we are doing is super exciting. Have a look. So I'm talking about a multi-class gallbladder disease detection from ultrasound images and radiology report generation framework. So this is what we are doing right now. We're collaborating with AIMS. We're collaborating with the radiologist of AIMS who has given us the report. So what I do here, currently in the previous models, I have shown you an image coming up, giving you a decision of yes, no, or a multi-grade decision, right? It will say cancer, not cancer, or it will say mild, moderate, severe, and no cancer. But here we are effectively not trying to do that. Rather, if you look at the pipeline, we will try the input of a radiology image in our app will essentially generate a comprehensive report the way a radiologist does. So it's like this. Please understand, when I look at let's say I am the radiologist, I look at a report, I look at an image. So my vision sensor has been activated, okay? Then my mouth is the LLM. My mouth is a large language model you can consider. I speak, and there is a visual cortex in my brain, which actually is the mid-level connect between my eyes and my mouth. Right. My eye, you just relate with the AI. My eye is the vision encoder. Let it be a CNN model or a vision transformer model. My mouth is an LLM. My mouth is like a ChatGPT or a Llama 3 or a DeepSeek, something like that. And my brain, the visual cortex, which is responsible for the vision thing, is a mid-level connect. So in my machine learning lingo, this is a trainable parameter. My mouth is a trainable parameter. My eye is a trainable parameter. And my brain is also a trainable parameter. So independently, I am having 3 trainable parameter networks which I can train. Please try to understand the procedure, how I'm trying to replicate that. I'm training a vision encoder, I'm training a mid-level connect, and I'm training an LLM model. My mouth can be considered as an LLM. And if I can train all these three, if I can train all these three, then I must be in a position to do the entire pipeline. So here what I am essentially doing is getting the radiology images and keeping the existing reports for these radiology images, making the computer understand for this radiology, this is the the report generated, so that the machine keeps learning just the radiologist tells, and then for unseen and new test images, it can generate reports on your own. Similar story for, this is actually the same story for we did for this one for rectal cancer also. This is also an ongoing work, but this is in more advanced stage, where you have used, you can see, let's say I use a CNN model or a base transformer model for the vision encoder, and I can use open sources like Llama 3. I think DeepSeek has also released it. I think all of you, the Chinese company has released a LLM called DeepSeek last week, DeepSeek R1, which works on reinforcement. So we are in quite advanced stage over here. Now here what we have done for rectal cancer database, we can input a rectal image and generate the entire, we call it explainable AI. As a user, if the model says that yes, you are diseased, you may not be happy. But with the report, if it generates, yes, you have a gallbladder stone and it is 5 mm, it is left aligned, it is cone shaped, it is blocking the urinary passage, something like that. If your detailed report is generated, you get more happy. And this is called as explainability, adding an explainability to AI. And the world at current state, if you're a cancer biologist or whatever field you are, unless and until you are doing an explainable AI app, most likely you will not be accepted at the current time, because binary decisions or multi-class decisions will not be accepted anymore. So one more, skin cancer, maybe this is the last one. So we also did a skin cancer, if some of you are interested in skin cancer. So for the skin cancer images, we trained a model. I'll not go to the pipeline, I think I've already crossed my time. So this is the pipeline. Once again, what we used here is that we used a dual autoencoder approach to regenerate images to ensure how the attention models can understand the significant or the discriminatory portions from that. The results are very good. You can read more about it. I think it is published in scientific reports in Nature. So these are all open source. So I would thank you so much. I have covered some of the cancers which we have done. And thank you. So I already overstood my time. So apologies. So thank you so much. If you have a question, you can ask me.
Please sit down. Please sit down. Please sit down, students. Are you listening? You have a class of sociology. You are? Any questions from the session? It was a very interesting session.
So I have a couple of questions.
Yes, please. Yes, please.
The first thing, in the early part of your talk, when you're talking about the predictive models, and you have used around 1600 samples. Now, I work with clinicians, and what I have learned from them, that a cancer or a diagnostic feature, if it works in North India, it will not work in South India. Okay, and it's just purely because of the genetics or cultural diversities and things like that. So, do you think this model can, predict on a global scale, or it needs more training, on different scales to, generate better models? Can this be taken to the next stage? My first question. And the second question is that when you talk about different models of image-based scanning and doing this, so what I learned is that the CT scan or any imaging-based instrument are now coming with their AI-based models to generate a report, and then doctor just sees and says yes or no. So where do we stand in that? Just some comments on this would be interesting to know.
Okay, so thank you. First question, as you asked, whether it's going to work in North India or South India, I have no idea because I'm not a clinician. I cannot say, but as a data scientist, what I can tell you is that your concern is absolutely right. We, in our language, we call it as intra-class diversity. For example, if you train the model to detect an Indian dog and you ask it to detect a German Shepherd, most likely it will not detect that German Shepherd as a dog. So that's how we look at, we call it as intra-class diversity. I do not know. If you ask me, I'm not a clinician, but your question is logical, and apparently it seems that it may not work. If you say the data interpretation or the data pattern or the data dependencies are different, so it may not work. You're absolutely right. So recommended is that we should actually use all possible data from South India, North India, everywhere to train our model. Yeah, of course. So that is understandable. And second question, you said that as all the equipments are coming with AI model, that is absolutely true. AI models have been scaled down to so much of low parameters. Even if a 5,000 rupees processor, you can process it in the current times. Okay, you have got NVIDIA Jetson Nano or Raspberry Pi, small chips will cost you 5,000 rupees. So even there's a 1 crore rupees instrument, you can always put a 10,000 rupees chip directly, and most of these AI pipelines are free. They're open source. You can deploy it and put it. So it makes complete sense. And of course, 5 years from now, maybe 2 years from now, every machine will come with an AI model. There's no doubt about it because there is no extra cost involved. A small processing unit can do it. Let's say you have an instrument of 5 lakh rupees. Nobody will mind putting a 5,000 rupees processor in doing that analysis. So that's what I'm not a clinician actually.
Thank you, Dr. Datta,
Yeah.
for the insightful presentation. I would now request the session chairs.
Yes, please.
Say ultrasound images, MRI images together. How easy or difficult it is to build such models when you have data from images, when you have data from socioeconomic parameters. How do you integrate and how do you decide what weight to put on which parameter?
No, see, currently whatever models going on across the world, they're all multimodal. If you talk about, let's say, you want to do a depression detection. So in depression detection, you're looking at the face image, the expression, you're looking at EEG brain signals. Okay, you're looking at other parameters also. So if you ask weight assignment and all multimodals, see, they're all independently trained. Just as I mentioned, a vision encoder, we are going to use a vision encoder for training the vision part. We can use a time series, a time series one-dimensional CNN or an LSTM or any dependency model like BiLSTM or attention network to do that. So they can be trained. And if you ask the weights, I mean, all these data patterns are different. See let me explain to you like this. When I look at you, let's say I look at your face. Your face is a two-dimensional static image. Your face is not going, the dependencies are not going to change. Okay. For this, you use a CNN model or a visual transform model. What you say when you talk, right? It's a sequence. So it has got a dependency. The previous word and the next word and the next word is a dependency. For that dependency, you use, of course, you're going to use a sequential model like a RNN or a LSTM or a BiLSTM or attention network. So whenever you integrate different categories of data points to train our model, we use different, training patterns. Because sometimes, look, I think you understood, some data points are specific point dependent. For example, your face, the points are fixed. It's not going to change. But what you speak is a text, which has a temporal dependency. So for this, you use a different. How to do that is once again a trainable parameter, which you can train. Okay, and they are very much doable. I mean, if any of the student or anybody is thinking that I will do only vision and leave in the current AI, you simply cannot, because decisions are not based on single pattern of data in the current times. And the subjects are coming up, multimodal AI, and that is buzz. I mean, if you use only one parameter, for example, these papers are little, 2-3 years back, but currently what we are doing, we're using everything. Whatever a doctor looks into. Yes, you have a question? Maybe the other speakers also have some.
We have to move for the next session. Hello.
Last question.
Yes, sir. From your last slide on the current ongoing project that you've mentioned, in which you're taking all the parameters, LLMs and vision and everything. From my current knowledge, I know for a fact if you want to benchmark a vision AI is still possible. How would you benchmark an LLM?
What benchmark means?
Basically, if we have developed an LLM, how do we prove that it's better than the other LLMs that are on?
No, we are not. We are not. See, at your stage, you cannot build an LLM. You know how much it takes to build an LLM.
Even if we have an LLM, how do we benchmark and tell which one would be more appropriate?
You already have foundation models like Llama 3. We are working on Llama 3. Llama 3.2 is our foundation model. So it's an open source where you can open the layers and train your things. Okay. Did you hear about the DeepSeek model which has come up recently? Do you know how many machines they have used? 50,000 H100 cards. I mean 50,000 boxes of supercomputers they have used to train that.
Like 1% of the US models that they.
See, building an LLM from the scratch is something which we cannot compete with the big giants. You can only work on the application level.
Okay.
Take an open source, take DeepSeek or take Llama 3. Okay. Or if you can afford $200, $500, you can also use OpenAI because they are charging money, and you build on it. You should not. I mean, you cannot compete. I mean, I don't know if you are super rich. I don't know, but it'll cost you some $500 million to train these kind of models.
Thank you. Thank you very much. So we have to move next sessions.
Before we move on to the next session, I would request the session chairs to felicitate Dr. Datta. Yeah, before we move forward, I just wanted to confirm the presence of Dr. Sunil.
So next speaker, Dr. Shima Bhatnagar. She is the Assistant Director and Professor, Amity Institute of Biotechnology. Dr. Bhatnagar has demonstrated history of research in mediclinical chemistry. It's a very interesting talk, and she is skilled in organic synthesis, drug discovery, drug design, and protein chemistry and biochemistry. Ma'am, we'll listen your triple conjugate definitely. Thank you.
Hello, Momin sir, आवाज आ रही है?
Everybody. I thank the organizers of the Global Cancer Consortium who have given me the opportunity to present my work to past few years now for synthesis of bioconjugate. I will be sharing some of the results of this study. So breast cancer, as we know, is one of the leading cancers in India. It is the number one cancer in females. And Globocan 2020 has predicted that countries with low and medium human development index have the greatest possibility of developing relative increase in cancer by 2040. If we look at the incidence and the mortality rate of breast cancer in India, we find that the incidence is 26.6 per lakh females, and the mortality is 13.7. Almost half of the females who develop breast cancer are likely to have fatal outcome of the disease. So the statistics are quite alarming. If we look at the state-wise age adjusted rate, we find that many states in India are having a very high AAR, and among the highest is Kerala, Punjab, Andhra Pradesh, Karnataka, Telangana, and these are statistics which were presented in the Lok Sabha in 2022. And many of these states have an AAR of more than 40. And Kerala has the highest AAR of 45.7. 17 states and union territories reported a higher incidence of AR in India's average. So the statistics are very alarming for India, and there is a need to develop drugs that could target and serve the Indian population. If we look at the breast cancer molecular subtypes, there are various subtypes and there are drugs which are available for various subtypes. We have the luminal A, which may be ER or HER or PR positive. We have luminal B. We have the HER2 enriched subtype. We have the normal-like subtype also. But there is a very aggressive type of breast cancer which is known as the triple negative breast cancer, which is basal-like. It does not have the hormone receptors, does not express ER or PR, it does not express HER2, and is it is highly aggressive cancer. So the triple negative variant is highly aggressive. It does not express these receptors at all, and because of this absence of these markers, there is the possibility of rapid metastasis and treatment resistance that may develop due to this disease. I would like to draw your attention to this statistics that was published in 2020, and we show that this is not our publication, but they show that TNBC incidence is 25.7, and the most alarming statistics is that women of younger age, that means between 46 to 49 years, they are suffering. That is the mean age for TNBC, compared to non-TNBC breast cancers, where the age is between 49 to 52 years. So, which means that younger females are suffering from this aggressive disease, and there is a need to address this, especially for the Indian population, which is very different from the Caucasian population. If we look at the treatment options that are available for TNBC, we have taxanes and anthracyclines, which are given to patients. Doxorubicin is one of the first line of treatment that is given. Doxorubicin intercalates, its mechanism of action is that it intercalates with the double-stranded DNA and breaks it. And it also produces reactive oxygen species and leads to oxidative stress, which is also responsible for the high systemic toxicity of doxorubicin. So how can we address these issues? Because we have a very aggressive cancer, we have a drug which is highly toxic, and if we want a female to take this disease for a long period of time, it may not be the disease that is leading to a bad outcome. It may be the toxic side effects which are actually leading to this outcome. So in our lab, we have been exploring smart drug delivery systems. We are looking into preparing a drug conjugate which could deliver the drug at low concentrations, and smart drug delivery systems allow low drug concentrations to be administered effectively at low intercellular concentrations. Therefore, they expand the therapeutic window of the anti-cancer drug. These smart drug delivery systems have improved specific localization, patient compliance, reduced toxic side effects, and controlled biodistribution. Many of these smart drug delivery systems, they are decorated with ligands, and they may be covalently conjugated with anti-cancer drug, or they may encapsulate these drugs. So once the drug is internalized, stimuli response inside the cell will lead to the release of the drug inside the cell and lead to drug action and apoptosis. Some of the targeting ligands that have been used for smart drug delivery include folic acid, biotin, lactose, to name a few. And most of the anti-cancer drugs, there have been studies which have explored their delivery through these smart drug delivery systems. So there are various types of smart drug delivery systems. We may have antibody drug conjugates, we may have quantum dots, liposomes. In our lab, we are exploring small molecule drug conjugates, and these may respond to stimuli such as pH, ultrasound, light. They may respond to enzymes. So there are different ways by which you can achieve the release of the drug inside the cell. Now, how does a small molecule drug conjugate achieve its selectivity? So the answer lies in the morphology of a normal cell and a cancer cell. In a normal cell, usually the receptors are expressed on the apical side. So a small molecule drug conjugate will not target the receptors if it is given to a patient who does not have these receptors expressed towards the bloodstream. But once the cells become cancerous, there is a loss of intercellular junctions, and the the morphology of the cells changes in case of cancer, and these receptors become exposed to these SMDCs and can be trapped through the receptors to be internalized inside the cell. So with all this premise in mind, we chose folic acid as one of the ligands that we wanted to use for the delivery of anti-cancer drug to the cells. So I'm just presenting a part of the data. Just to achieve the delivery of the cells, we also chose curcumin as one of the chemosensitizers, and we first wanted to determine whether we can achieve the delivery of curcumin inside the cells expressing the folate receptor. So if we look at the structure of folic acid, it comprises of three parts. We have the pteridine unit, we have the para-aminobenzoic acid unit, and we have the glutamic acid unit. Now, the binding of folic acid inside the folate receptor happens through the pteridine unit. So that part of the molecule is not available for any type of conjugation because that will affect its binding to the receptor. So we have the alpha carboxylic group, we have the alpha carboxylic group, and we have the gamma carboxylic group, which we can use for functionalization. And we have used simple conjugation techniques to be able to functionalize this and prepare a conjugate of folic acid and curcumin, which is stimuli responsive and can release the curcumin inside the cell. So this is the first part of the study where we synthesized this folic acid curcumin bioconjugate. We used DCC NHS and prepared the folic acid NHS ester, and we reacted it with curcumin in the presence of base to get the folic acid curcumin conjugate. We looked into the structure of the compound spectroscopically, and we have the mass data, which is in agreement with the experimental and the calculated values. The UV visible spectra was also very much was matching with folic acid and curcumin, but there were slight variations because conjugation had taken place. The IR of the compound, we compared folic acid, curcumin, and our conjugate, and we find the ester linkage at 1747. The carbonyl of curcumin 1627 was retained in the conjugate, and folic acid 1696, the other carboxylic group, was coming at 1666 in the conjugate. Additionally, we also looked at the NMR of the compound, and the characteristic peak at 5.8 ppm for the methylene group of curcumin, and the methoxy groups of curcumin indicated that the compound was ready. We also looked into the HMBC of the compound, where we compared the proton NMR with the C13 of the compound, and we got cross peaks in agreement with the structure that we were proposing. So we went on to study the biological activity of the compound and the effect of curcumin and folic acid curcumin conjugate on two cell lines, MCF7 and MDAMB231. MDAMB231 is the accepted model for studying folate receptor studies. So we have used this as our model. When we studied the MTT assay for free curcumin, in MCF7 our IC50 was 31.8 micromolar, and MDMB the IC50 was 38.6 micromolar, and this was very much in agreement with the literature values that are reported. Our conjugate, on the other hand, had a very low IC50 that was 19.46 microgram per ml, and in MDMB it was 12.83 microgram per ml. We looked into the cell proliferation assay, and we chose two concentrations for curcumin and folate curcumin conjugate. So curcumin 15 micromolar and 30 micromolar, and folate curcumin which at 5 microgram per ml and 10 microgram, much less than the IC50 values. And we find that there is a change in the morphology and the number of cells in MCF7 as well as MDMB cell line after 24 hours of treatment. Next, we examined our conjugate in comparison to curcumin, and we did the cell cycle studies, and we find that in MCF7, after at these concentrations, 15 micromolar and 30 micromolar of curcumin and 5 and 10 micromolar of the microgram per ml of our conjugate, we find that the cells are getting arrested in the G2M phase. This was seen in the MCF7 cell line and also MDMB cell line. A greater proportion of cells were found in the G2M phase, suggesting that there is a cell cycle arrest. Next, we did the further studies on these, studying the apoptosis, and we find that untreated cells are, when they are treated with curcumin or the folic acid curcumin conjugate, the cells move to early apoptosis, and in MDMB, from early apoptosis, they moved directly to necrosis, did not go to the late apoptotic stage, suggesting that our conjugate was working. We also did the in vitro cell migration assay in MCF7 and MDMB. Scratch assay was done, and we find that cell migration was inhibited in the presence of the conjugate, as is evident from the pictures. In both the cell lines, we are seeing, so this indicates that the cells are not able to migrate when they are treated with the conjugate. So keeping in view the theme of the session today, I am also presenting some of the work which we did under my Union of International Cancer Control project, where we have used AI platform again for anti-cancer drug discovery. So briefly, I will be also talking about that. So in this study, we initially started with the plant Clerodendron importunatum, and we did the fractional extraction, which was polarity based. We prepared 4 extracts: hexane, chloroform, ethyl acetate, and methanol. And we studied its anti-angiogenic activity using the CAM assay. CAM assay is a fertilized chick assay where you study the development of the CAM after treatment and before treatment with the plant extracts. And we found in our studies that our ethyl acetate extract was most potent compared to the other extracts. Now, when we started quantifying these membranes, we found that there is no specific software which is available by which you can quantify. So we wanted to know if we could use AI technology and also look into quantification of this membrane, and that's how the project started. And then what we did was that we used retinal vessel segmentation data. We used deep learning, and using this model, we studied our CAM membrane using retinal fundus images. So in this panel, now we used 2 types of CAM images. Some of them were generated, the in ovo images were generated in our lab at Amity here itself. And the ex ovo images we got from our collaborators in Turkey. So these were independently obtained, and we we wanted to study whether the results are matching not only with our results but independently obtained images from other collaborators. So as we see the ethyl acetate extract, here you see that the vessel formation is very much affected post-treatment, and we annotated this data and looked for branch points and endpoints. We identified 16 quantitative descriptors and prepared a heat map and wanted to compare our different extracts and whether they were the vessel formation was in agreement with our results that we obtained previously, that our ethyl acetate extract is very potent. And indeed, using the retinal fundus images data set, pre-trained data set, we could find that our ethyl acetate extract at 100 micromolar was indeed very active, and we could see a lot of activity in this case. Similarly, the results obtained for the Turkey data set also were in agreement with what results were obtained by the research group independently. So lastly, we did a number of other spectroscopic studies, and we found that in the ethyl acetate extract there are some peaks. The NMR indicated that there were some peaks for compounds which may possibly be flavonoids, and so we collaborated with our colleague in at Agarkar Institute, Pune, and we looked into the LCMS profile and compared it with the NIST data. And I'm happy to share that we have some very new compounds which have come through the study, and we will be taking the study further based on the data that we have obtained for the LCMS. So these are some of our publications and patents and books related to the area that I have presented today. And no work can be done alone. So I have most of my work is collaborative. Of course, Dr. B.C. Das, we have done collaborative work on the triple conjugate, and I have just presented the dual conjugate results today. Dr. Sarachan Janga, he is at the Luddy School of Informatics, and we were working together for our AI work on drug discovery. Dr. Nidhi Srivastava, she is the expert in the CAM assays. So the CAM assays were done by her, and the further imaging assays, we took the pictures of those assays and did the AI experiments in US. Dr. Naside, she is from Turkey. She has provided us with the X-over experiments for CAM. And of course, Dr. Pratibha Srivastava was involved with the LCMS analysis. So many thanks to her. Of course, the students are the backbone of the lab. So Mr. Abhilash Rana, Praveen, Abhilash was working on the folic curcumin conjugate. Praveen was working on the clerodendron project. Prabhat is a biologist from IMSR in Dr. Das lab. He's working for the biology experiments for our conjugate. And Hunter Gill is from Indiana. He also worked on our AI experiments that we had done. I also thank Indian Council of Medical Research for funding our research on the folate project, and Union of International Cancer Control that funded the study grant for our work on the AI project. Also, many thanks to the organizing committee for giving this opportunity to present our results.
Thank you very much. So it's open for the questioning. Any questions from the audience? Okay, sure.
So if there is no question, I will ask you, Seema. Very nice presentation I have seen. And the priority which you have taken to make the triple conjugate with the folic acid and with this one. So have you tried any conjugate with the tamoxifen also?
Yeah, we have a number of conjugates that we have prepared. We have, here I have just presented the ester linkage. We have also explored self-immolative linkers like azobenzene linkers, so that when it goes inside, when the conjugate goes inside, in the presence of the glutathione system, it will just self-immolate and release the drug. And we have done some aspirin tamoxifen conjugates. We have also done some genistein tamoxifen conjugates. So we are basically the USP of all these conjugates that we are preparing. We are using natural products as the chemosensitizer that will facilitate, not only give the anti-cancer activity, it will facilitate the entry of the anti-cancer drug at low concentration, and it will also take care of the side effects, because most of these compounds modulate the activity inside the cell. So the toxic side effects can be taken care of, and that is evident from the IC50 that we are getting, which is at low concentration. And since these conjugates are 1:1, they have been there's one molecule of this and one molecule of this, so that's why the concentration is really, really less, but the activity is better.
So they don't need any kind of carrier like HP beta CD or dendrimers to.
No, the folic acid is the carrier.
Okay.
Because
Itself it works
yes,
as a carrier.
TNBC cells overexpress a folate receptor.
Okay.
So folic acid is the targeting moiety which will take your compound to the cell
Okay.
where it has to be delivered. And in rest of the normal cells, folate receptor is expressed on normal cells also, but they are apically expressed, so they will not be able to capture the drug conjugate.
Thank you very much, Seema.
So I think excellent talk, ma'am.
Thank you.
Definitely.
Please, go ahead.
So I think partially you have already answered my question on the folate part, because normal cells also express folate, but it is overexpressing on the apical part. That's the main point. And obviously these two components like curcumin and the drug, they are actually requiring in the lesser amount when
Yes.
in the, so definitely they will have a synergistic effect. I mean inside the cells.
Yeah.
So maybe you can have a intercellular dynamics. You can look into confocal or some intercellular changes. If you can look into it, and you can say add a new layer of information there.
Yeah,
actually.
sure.
What is happening inside the cell?
Yes,
like,
yes. This is work in progress.
Yeah.
Of course, I mean, it takes a lot of time. Many questions need to be answered, and its entry into the cell.
Yes.
But we have some studies where we have seen curcumin itself fluoresces. We have seen some of the studies where we have seen curcumin entering the cell through fluorescence. Also, those studies I have not shown here, but using other dyes we could also do that.
Yeah. Sure.
Curcumin itself fluoresces. So we have
Yeah.
just looked into that aspect.
Thank
We
you.
could use other dyes.
Ma'am, I'm just curious to know, you are adding folic acid, right? Now this folic acid is also a vitamin. Okay, now the thing is that, my clinician says that when they treat cancer, they don't try to give vitamins because that also boosts cancer cells. So they give drugs once they see the, the efficacies of the treatment is good, then they help by giving vitamins to, boost the normal cells as well. So the question here is that, maybe that could be one of the reason why you're getting low IC50.
So as I said, sir, these are stimuli responsive. So it's not folic acid which is entering the cell. It is cleaving in as soon as it endocytosis happens. The acidic milieu of the compartment will cleave the bond. That's why we have chosen bonds which can be cleaved at low pH. So when they will cleave, folic acid is not entering. It is curcumin and the anti-cancer drug which is entering the cell. And on top of it, why I have used folic acid is because folic acid is overexpressed on the cell. On any TNBC cell, there is overexpression of folate receptor. So we are just using it as the targeting moiety where we want to target. It should not go to it. Right now, the toxicity that you see for any anti-cancer drug is because it is non-specific. It targets all the cells. So that's why you see more toxicity. But we don't want that. We want it to be more specific. And similar example is vinblastine. There are many examples. The industry is already working on it. So there's vinblastine where they have used folic acid and vintafolide. Now it is out of phase 3 trials. But here the USP of our product is that we are using curcumin. That is the additional thing that we are using. And curcumin, we know that it is a wonder molecule. The problem with curcumin is its bioavailability. So we are addressing all that through the conjugation chemistry that we are doing, so that it is able to enter the cells. It is more bioavailable, and it also interacts with all the pathways that I have shown in one of the slides, and also imparts some anti-cancer effect, chemosensitizing nature, many things it can do together synergistically with the drug.
So
that mechanistic part which downstream targets, because folic acid and curcumin both are going inside.
Yeah.
So what which particular particular target? Because curcumin are multiple levels. It targets many pathways.
Yes, yes.
So have you gone through that?
No, those we have to still do. It's a very long study which may be required for this, and of course funding is always required for doing more studies. So that is definitely something we have to explore, that what happens once it enters. But our first question is it should enter and it should show effect. So right now we have the proof of concept that it is entering, it is also showing effect. Now we need to do further studies inside the cell. So markers and all those things we will be doing.
one quick question. There are a lot of talks on this breast cancer and posters as well. I was unable to understand or find the statistics as to in India, what is the percentage of ENBC, what ER positive, HER positive, what is the percentage, do you know?
No, I showed that slide 25.7. I mean, it's very high. TNBC is higher in India as compared to Western population. We are seeing more incidence of TNBC in Indian population. And we need to, most of the drugs that first line of treatment, you will give tamoxifen, or you'll give all these chemotherapeutic adjuvant, you may give tamoxifen. So what we are seeing is that those drugs do not cater to the Indian females. We need to have our own drug which we can use. There must be. There must be.
Thank you very much. So just little announcement, because after that, it's very nice talk. So yeah, you can go ahead.
Thank you, Dr. Bhatnagar, for the wonderful presentation. Now I would request our session chairs to felicitate Dr. Bhatnagar, please. I would now request Dr. Shrudeep and Dr. Archana to felicitate our session chairs.
Okay, this last announcement. Actually, new speaker, Dr. Shunil, actually not well. So he will deliver his talk in the I2 moot court. So I am requesting all the audience to move to the I2 moot court. So all remaining talks for today before lunch will be continued over there. Thank you very much. Thank you very much. So officially we are closing the session from F3-Seminar hall. Thank you very much. Thank you everybody.